Thank you for the opportunity to show my data here. It's a great pleasure. Well, as a renal physiologist and nephrologist, I'm interested in studying ion transporter in renal tubules. I will say a few words to justify this investigation. And after filtration of huge amount of sodium in glomeruli together with the counter anions, mainly chloride and bicarbonate, renal proximal tubule must reabsorb almost 70% of the sodium and water filtered in glomerulus. It corresponds approximately to 17,000 millimoles of sodium a day, and almost 11,000 millimoles of sodium is reabsorbed through sodium proton exchanger, the isoform 3 or SLC9A3. Well, NAD3 is a very abundant protein at apical membrane of proximal tubules, especially in the early segments. Parathyroid hormone was one of the first hormones described as modulator of the fluid reabsorption in proximal tubules. Besides inhibition of the sodium phosphate cotransport, it also inhibits sodium proton exchanger. Acutely, PTH induces phosphorylation of NAD3 by pKa, what triggers its retrieval from the apical membrane by endocytosis. In experiments of our lab, we have verified that PTH reduces the NAD3 protein and NAD3 mRNA in rat renal cortex. And these animals were treated with high doses of PTH by subcutaneous implantation of slow release PTH pellets, and another group of animals were parathyroidectomized, and the kidneys were analyzed after 8 days. And we observed, sorry, what I did, okay. We observed that, where is the? Okay, okay. In excess of parathyroid hormone, we observed a decrease in NAD3 protein and in NAD3 mRNA. Without PTH, we observed an increase in NAD3 protein and in mRNA. The effect of PTH on the proximal tubules, we studied also the effect of PTH in a proximal tubule cell line from the opossum to Delphis virginiana, OKP cells, because these cells are commonly used to study this NAD3 and sodium phosphate cotransporter. And it was our intention to analyze the promoter, so we need a cell line. So in these cells, we observed that the treatment of PTH induced a decrease in NAD3 total protein after 16 hours, and the decrease was higher after 24 hours. And there was also a decrease in the surface NAD3 protein, biotinylated NAD3 protein. After 6 hours, we can already observe that this decrease in the apical NAD3, apical or surface NAD3. Okay. NAD3 mRNA was also decreased after 24 hours. And we have observed also a mild decline in the NAD3 mRNA stability in cells treated with PTH for 24 hours, but we did not investigate further this phenomenon. Well, we decided to analyze the promoter. We tried to obtain the opossum virginia promoter region of the NAG3 gene by PCR using the PCR vector technique, but without success. So we decided to use the rat NAG3 promoter in OKP cells as done by several researchers in this area. It's the most common. So with the rat NAG3 promoter, when we analyze the proximal promoter, we can identify several SP1 and EGR1 and Sp3 transcription factor binding sites, and it was previously reported that these transcription factors are important for to modulation of NAG3 expression in several conditions. For example, the use of PMA that increases EGR1 expression and affects the NAG3 expression. And in other condition, Sp3 and Sp1 and Sp3 is highly increased in proximal tubule cell line in culture when they are confluent and forming dominant cells. So we analyzed this rat NAG3 promoter in OKP cells, and well, we analyzed much more, a lot of different segments from 2,000 base pairs and different segments, but we chose this proximal promoter because the effect of PTH were more clearly observed in the proximal promoter. Anyway, at least 44 base pairs of the 5' flanking sequence is required for effective expression of the reported gene, in this case a firefly renira luciferase. Firefly luciferase. Although with 25 base pairs, we can also observe transcription above the basic level. As we analyzed the effect of PTH on rat NAG3 promoter in several segments, but the effect was clear in the proximal promoter and core promoter, and this effect was sensitive to PK inhibitors, was reverted with PK inhibitor KT5720, and are sensitive to inhibitors of JAK and STAT kinases. PTH1 receptor is a G protein coupled receptor which activates adenylate cyclase and increases cyclic MP and activates protein kinase A. So it's not a surprise that the effect is inhibited by protein kinase A inhibitors. And one thing that JAK is a tyrosine kinase that activates multiple transduction pathways, including activations, the cytokine glucoprotein 130, GP130. And relationship between PTH and GP130 was described by Stendhal, that reported that the osteoblast induced, the PTH is induced osteoblast differentiation requires GP130. So, and GP130 recruits STAT. Maybe this inhibition is related to this pathway, but we have to analyze this. Well, we realized some punctual mutations in the Sp1 and Sp3 EGR transcription factor consensus binding sites in the rat NAG promoter and G changed by to A, another G to A, and CG to AA, CG to AA. And all these mutations abolished the effect, the inhibitory effect of PTH. And we even observed increase in the promoted activity in presence of PTH in presence of the inhibitors. As we have previously observed with the inhibitors, now there was an increase in promoter activity in presence of the inhibitors. Well, considering these observations, I decided to analyze the expression of the sp3 protein in OKP cells treated with PTH. sp3 protein shows a very complex expression pattern, and we observed these several bands, and here I show Western blot in HEK293 for comparison of the complexity of this expression pattern. And we observed that PTH induces increase in the sp3 long isoform 1. These isoforms are apparently derived from different transcription start sites and to post-translation simulation of some isoforms. That's why we have so many bands. And sp3 long isoform 1 was increased by PTH. The long isoform 2 was decreased. The short isoform 3 was increased, while the short isoform 4 was decreased. And these two bands apparently are short simulated isoforms and was clearly increased in presence of PTH. There is another band that was previously reported when cells were lysed with SDS, and we detected also these bands, and they were this band was increased in presence of PTH. So these modifications were significant. So long isoform increased, 1 increased, long isoform 2 decreased. The short isoform 3 increased, the short isoform decreased, and the simulation was increased. Well, we did not observe any change in sp3 mRNA level. EGR1 protein was not changed in presence of PTH. So this is Western blot. It was a single band, and no difference in control between control and cells treated with PTH. On the other hand, we observed an increase in EGR1 mRNA. This mRNA was measured by real-time RT-PCR. After that, we did electrophoresis mobility shift assay with fragments of the rat NH3 promoter, -6 to 1, from -6 to 1 to -42. That includes sp1, sp3 binding sites, and that are EGR1 binding site. And we observed that a shift of the probe with EGR1 antibody. We were not sure if the sp3 antibody is not clear. That there is a difference, but it's really not clear if there is a difference. Maybe there is it decreased the amount of the in this position. And we use another probe from -54 to -24. That includes also sp1, sp3, and EGR1 binding sites, but it was also sp3 did not change, and antibody for any sp3 did not change the probe migration. Considering these results, we propose that we need to clarify this, but that PTH changes as PTH changes sp3 expression of KP cells. Maybe sp3 may inhibit NAG3 expression by itself. So there is Valin and Gill observed that there's the inhibitory effect of the sp3 transcription factor, and they proposed at least two models for transcription inhibition. One of them is sp3 summoylation might recruit a transcription repressor, and this transcription repressor could induce closed state of chromatin that hide important binding sites for basal transcription factors, and it will making possible the binding of the RNA polymerase 2 and the other factors. So the transcription is inhibited. Alternatively, it could change, interfere with the paused RNA polymerase 2 to productive elongation polymerase 2 that depends on the positive transcription factor B. This positive transcription elongation factor B normally is maintained in an inactive state in a complex with a small nuclear RNA with other proteins like LARP that interacts with the 3' of the 7SK small RNA, and EXIM that interacts with the PTFB, and this protein that is responsible for the cap of the 5' end of the 7SK. PTFB is a complex which contains CDK9 kinase and the cyclin T that modulates CDK9. And EXIM interacts with cyclin T and inhibits the complex. Well, the switch from pause PO2 to elongation PO2 depends on the liberation of PTFB. Different signaling could be that, and PTFB will be released from this complex, and it induces phosphorylation of some negative regulatory factors as NELF, that negative elongation factor, and the DRB, sensitive inducing factor that are repress the transcription. And in this state, the RNA polymerase 2 is in a positive state. When PTFB is liberated, it phosphorylates both the DSIF and NELF, and the NELF is released from the complex, and PTFB is incorporated in the transcription machinery, and the DSIF acts as a positive elongation factor in this complex. So there is a transition from paused RNA polymerase 2 to elongation RNA polymerase 2, where sp3 recruits phosphatase, which removes the phosphate added by PTFB. So maybe some PTH signaling could induce this sp3 modification that interacts with a phosphatase as PP1, and it could dephosphorylate this negative regulatory elements, and sustain the RNA polymerase in a positive state. So our hypothesis is that the complex sp3 pp1 might influence the balance between paused and elongation polymerase 2. We don't know which is the role of the EGR1 in this process. So we are working on it now. So we have to elucidate these questions. Thank you for your attention.
Do we have any time? No, I don't think
Not
so.
really, no.
I want to thank you, Dr. Reboucas, for a very interesting talk. Could you hang around for a moment? We have one of these for you. So, come on. And you are a hardy group of scientists.